So Georges, you're the VP Business Development of Celyad, a company in the market of the CAR-T and also in the cell therapy market. Maybe we can start by saying a word about your company, because so your company was known as cardio-free bioscience before, and you are developing a cell therapy for cardiology. So now you're entering the CAR-T. Can you just tell us a bit more about these changes in strategy?
Yes, so the company was incepted back in 2007 to develop cell therapy for ischemic heart failure. And we took the therapy actually from preclinical stage and licensed from Mayo Clinic all the way up to completing a phase 3, registration phase 3 in Europe. And so we went as far as possible with our cardiology platform in terms of development. Definitely we are not a commercial company, so we will be looking to partner this for the commercial phase. And the company starts thinking, that we are coming to an end of a cycle with our cardiology program, what to do next. So we've explored many opportunities, mostly in the cell therapy, because this is where we are legitimate and where we can leverage what we have developed throughout our cardio program. In terms of manufacturing, we have our own manufacturing facility. In terms of preclinical, in terms of regulatory and clinical development. So we've considered many options, and we came across the opportunity in CAR-T, and the technology was very attractive. The preclinical data were very compelling. The IP was very strong. So then we decided to move into this new area and to extend our pipeline outside of the cardiology into the, what just discussed, the immunotherapy for oncology.
Yeah. The CAR-T market is really attracting a lot of different companies because of this huge potential. We can mention Cellectis in France, or we can mention June Therapeutics or Kite. How do you differentiate from them? What's the difference with your technology?
Okay, so first, just to give an overview very quickly on what is the CAR-T. The CAR-T is actually the first time that was described, it was described in, I think, 83, 84. It was used to be called the T-body, meaning putting an antibody and a T-cell. And this is the CAR technology is basically transforming an antibody into a chimeric receptor that you will put on the surface of a T-cell. And all those companies that you cited and all the excitement and the great clinical data that were obtained with CD19 are basically transforming an antibody into a chimeric receptor on the T-cell. So what we are doing is we are not using chimeric antibodies, we are using human natural receptors from NK cells to transform them into a chimeric receptors and move them out of their NK environment into a T cell. So from this perspective, we are still working as a chimeric receptor, but not using antibodies to make up those chimeric receptors, but using natural receptors that are present on the immune cells. And this is totally different from what everybody is doing. And the IP, and from this perspective, is totally independent of all the IP that exist in that field.
Okay. Maybe we can mention also an important difference in the CAR-T market between autologous and allogeneic approaches. Can you tell us more about your approach?
So today, indeed, there are two ways of doing cell therapy. It's not specific only for CAR-T. It's either autologous therapies, meaning you take the cell from the patients, you ex vivo modify those cells under GMP condition, and you reinfuse it into the patient. So this has the merit of the safety, because you are taking cells out of the patient and putting them into the same patient. But it has some issues, or some, I would say, difficulties and challenges from manufacturing perspective. One patient, one batch. The allogeneic is basically transforming that by taking the cells not from the patient, from healthy donors, so you can then basically select the donors, probably profile the donors for many reasons or for achieving a specific aim you want to have, and then manufacturing multiple doses, and then put those doses into the patients. It has some own difficulty, but the nice things about it that from one batch you can then manufacture multiple doses, and then you can reduce your cost of goods. So those are the two approaches today. And basically Celyad is developing both of them because we believe we are kind of technology agnostic. Autologous will be the first therapy approved. This is what regulators are well accustomed to see, and from safety perspective is going to be the first generation. And the second generation that will follow up will be definitely an allogeneic. The allogeneic have some regulatory challenges that we need to overcome, us and many other companies that are developing in the field.
I checked your quotation, your market cap yesterday. You're listed on two markets, so Euronext and the Nasdaq in New York. So your market cap is around 448 million euros to be exact. Why did you choose to go to New York to raise money?
Well, I mean, we are a European company. We want to keep those roots as a European company. Nonetheless, immunotherapy or investors that are interested in investing in immunotherapy companies are mostly, or at least this is our understanding and analysis, are based in the United States. So definitely American investors can come and invest in European company listed on Euronext. But if you want to attract, the big players, start developing in big centers and develop your product also in the United States, it's very good to be present. So for us, it was a way of raising money in the right time frame, because this happened before the summer of 2015, where all the biotech actually went down and the market went down. So it was a good window for us to raise enough money. We've raised around 100 million, and it gives us now the opportunity to have all freedom to operate for at least 3-4 years to complete all our clinical plan. So we are not in a need for a partnership, so we can develop our pipeline as we wish, and give us a great freedom from this perspective. So definitely there are good funds to attract and to raise in the United States, where we felt at that time we couldn't have raised enough money in US.
And
in Europe.
Cellectis also did the same as you with double quotation.
Cellectis did the same, and other companies, European companies, also did the same. So typically we went for a private placement before like a priming round in the States, and the fund that has invested in our private placement and also invested in our IPO.
Now let's talk about your clinical trials. So CAR-T is really showing extraordinary results. We have seen Novartis with one of the trials having 93% of complete remission in children with leukemia. Where do you stand with your clinical trials right now?
Yeah, so indeed Novartis was its CD19 CAR, and Novartis and other companies, but Novartis is the most advanced, has reported a tremendous and very astonishing, I would say, clinical data in CLL and ALL. However, this doesn't cover the full leukemic patient. There are many other leukemic patients where CD19 did not show an efficacy. So from this perspective, there are other indication or other disease in liquid tumors where there are potential market for other CARs than the CD19. This is to start with. And second, the most today unmet medical need is also in solid tumors. And this is where also Celyad is looking to develop all its future clinical plan in solid tumors. And this is based on a very solid preclinical data that we have generated, where we have shown that our CAR-T has a distinct mechanism of action. It doesn't only kill the cells like most CAR-T does, but also change and modify the tumor microenvironment to actually recruit the immune cells, the activated cells, and actually repress all the negative T cells. So from this perspective, we feel we are very well placed to start addressing the solid tumors, and this will be quite differentiating development from many other CAR-T companies.
Are there other companies trying to target the solid tumors already?
Well, yes, there have been some failure in the field, but there are more and more companies looking to do that. The thing is that you need a specific tumor antigen to target specifically the solid tumors, but also, as was mentioned previously, you need to be able to infiltrate the tumors. The ligands that we are using for the NK receptors are broadly expressed in most solid tumors, so we can leverage on our preclinical and our safety data to go in multiple solid tumors. And the mechanism of action that we have demonstrated in the preclinical models support that our cells infiltrate and go into the tumors to actually kill the cells and recruit the immune system.
Yeah. So one last question about Novartis once again. For once, it's a big pharma which is driving or which is leader in this race. What do you think of their approach? What do you think of what they are bringing to this market?
Well, I mean, what Novartis is doing today in that perspective is very impressive. I mean, I was before being at Celyad, I was also VP Business Development at Cellectis, and when I was going out and trying to partner our CAR CD19 at that time, everybody was like saying, hey, you know, you are nice, but you know, keep enjoying, keep playing, but this will never work. So I think when Novartis did the deal with UPenn, actually this transformed the whole field, and this is what allowed many other transactions and IPOs like, you know, Juno and Kite. So Novartis is leading, and thanks to Novartis, it opened actually the field to many other transactions, like Pfizer also went into the CAR, Amgen. So and they still have today one of the largest pipeline. They have about 12 to 15 CARs that are in preclinical or ready to enter a clinical phase. So I mean, I respect what they are doing, and the field needs those kind of things to attract investors and to attract attention, and also for the benefit of the patients, hopefully.
Okay, so now we will take some questions from the audience. Yeah, any questions? I still have some questions. So I have another question about the patents. So you said this market is really competitive, so there is a lot of companies and a lot of technologies, meaning a lot of IP. Where do you stand in terms of IP? What's your strength?
So as I mentioned in the introduction, first the chimeric receptor we are using, we are not using antigen, and we are not making chimeric, you know, like the backbone that has been described. So we are using totally different approach, which has its own IP, and most of those IP today is granted by USPTO and or pending and outside of US. This is on the, let's say, the chimeric constructs. On the allogeneic technology, we have a patent portfolio that was filed way years before that all this hype in the CAR space. So we have two patents that are granted. One is a fundamental patent on the allogeneic, meaning describing how to generate a TCR deficient cells expressing a CAR, independent of the technology that is used to actually inhibit or modify this TCR. So this is basically applied to any allogeneic T cell, and this patent has been challenged and re-allowed. And today, you know, I mean, we are looking, if we'll be happy that will be challenged again to be re-allowed, which means this is a very important patent. If it was an important patent, nobody would care about. And second, we have another patent on a specific technology to make those T cells. So from this perspective, Celyad stands as independent of any other patents. Nonetheless, we are also open to discuss and to consider working in collaboration with others. And today we are in many discussions with pharma and biotech that they would like to access our portfolio.
Great. One question from the audience?
Probably I was clear. And there is a question.
Yeah.
Hi. You were mentioning the capabilities of your platform to attack solid tumors. I may be mistaken, but most of the liquid tumor programs right now target CD19+. So how do you open up in solid tumors other targets?
Okay, so because then I need to explain the biology of the receptors that we are using. So actually we are using receptors that are expressed on NK cells, natural killer cells. So those receptors actually by nature recognizes ligands that are expressed on tumor cells, and this is not limited to solid or liquid tumors. 80% of tumor cells express those ligands. So when you make a chimeric receptors of those NK receptors and charge them on a T cell, then this T cell is able to target and recognize most of the tumor cells. And so we are not relying on a specific antigen. So there are many ligands. So if one ligand is shed by the tumor to escape, then there are other ligands that are expressed. So the possibility of escaping or a clonal selection of tumor cells is basically not existing today. So this is how we address by using those natural receptors that target tumor cells, and the fact that there are multiple ligands expressed on almost every single tumor cells. This is how we can target both liquid and solid tumors with the same constructs.
If we have one question in the back.
Hi, just to continue on the technology, I think it's quite neat that you're taking the receptors from the NK cells and sticking them on the T cells. How does your approach compare to, say, Nantes Quest, who directly develop NK cells for immunotherapy?
Indeed, there are today many alternative technology to use NK cells, and the reason for that is that NK cells, they are very effective in eliminating tumor cells. And this is what they do in every healthy person. They screen for those malignant cells that appears and eliminate them immediately. And from time to time, there are cells that escape and the tumor forms. In our case, actually, instead of using, the problem with the NK cells is making them proliferate and grow them in large scale. So we overcome this limitation by the fact that we are supercharging NK receptors in T cells, so we can grow those cells and make them proliferate basically limitless. So this is the promise of the T cell.
And how does that affect the tox profile? So are they not a lot more dangerous than your traditional CAR T cells?
Okay, so again, then I need to explain further the way, because of that, the T cell we make, they don't persist for a long time, so they stay for a limited time in the patient and disappear. So what we are looking more is for a pharmacological agent than for cells that can proliferate and go uncontrolled in the patient body.
Okay, thank you, Georges.
I have also a question, a technical question actually. How do your receptors distinguish between healthy and tumor tissue or tumor cells?
Okay, those ligands are only expressed in stress cells, and by stress mean malignant cells that have DNA damage or cells that are inflammatory or infected with viruses. So healthy tissue does not express. Nonetheless, I just explained, though our cells persist or stay in the patient for a limited amount of time, so they don't proliferate and then expand in the patient. So if there are conditions that appears later in the patient life where those ligands are expressed for inflammation, let's say, then our cell doesn't exist anymore because they do the job, they kill the tumors and disappear.
Do you have a suicide switch in the cells?
We are considering adding that, but we don't feel at this stage that is needed.
Thank you very much.
Thank you.